think chapter 13 is looking at the experimental evidence, potentially. Like, how do you determine a lot of the nucleotide stuff experimentally? So what are you doing in the lab? So we had a similar chapter for our proteins that was some chapter earlier that we skipped over. I think it was chapter 5 or 6. So there's a similar chapter to that for your nucleotides. So what we're going to try to do today, and wherever I can backfill space, is to just cover some more information about the whole DNA replication, transcription, and translation, those kind of overall processes. So we'll talk about some of those aspects, but for the most part, I believe all of this information is already covered in your biology class. That was the impression that I got from last year's students, was that it was pretty much just a review, nothing really new or different. So this whole process overall is known as the central dogma, and it's just looking at the individual steps of the genetic information getting out into the cell, where you're generating your proteins and what the proteins can go through and do. Your general steps: replication, transcription, and translation. If you're anything like me, you keep getting translation and transcription backwards. Your replication is just looping within, so you're typically looking at DNA to synthesize DNA, or officially RNA to synthesize RNA. Then your transcription is that process of going from DNA to RNA, which can be reversed in certain cases. So we looked at viruses, retroviruses potentially doing that. And then there's translation, which is taking your RNA out to your proteins. Okay, so those are your main sections within this. One of the things that your textbook does is within each of these little chapters where you're talking about each of these individual sections is it also gets subcategorized or sub-subcategorized into prokaryotic processes versus eukaryotic processes. Okay. The eukaryotic is much more complex, so ultimately there's a whole lot less known about it. So for the most part, everything is explained through the prokaryotic sense, with some kind of adventures off into the eukaryotic differences. Okay. So we'll see these as we kind of go through it. To start with, we're looking at our replication process. So one of the first things that comes up as a famous experiment is that when we go through and we replicate DNA, do we get completely new DNA, or is it only semi-new? So what I mean is we have a couple examples. You could look at the front or at the back. So in the front, we've got those two red parent strands. Okay, what those are supposed to represent is the starting DNA. So what this question is asking, okay, is it completely new or semi-new? So if we're saying completely new, when we now go down to look at our first generation, so in this case we've gone through and done one replication cycle, we end up with two new or two sets of the same genetic information. Okay, so if it's completely new, what's happening? Well, we're going to take all of the pieces of our old, and we're going to take some new pieces, and we're going to intermix them all the way through. Okay, so if we looked at our genetic information, we would expect to see red, then blue, maybe a bunch of blues, and then a red. You'll notice that I'm drawing those all in the same chain. Okay, so everything is just kind of getting intermixed. Okay, or are we going through a semi-new process? Okay, we have to have some new because that's how replication occurs. You're making a second structure. The question comes is, what do we end up seeing out of that? Okay, and there's a couple processes. One, we could go through, and we'd have, that's my attempt at double-stranded DNA, our parent strand, and then we would have, again, my attempt at double-stranded DNA, okay, the daughter cell. Okay? Or do we end up with a process where that parent strand is evenly mixed? Okay, so we're looking at these two systems. So the first system we discussed doesn't really make any sense to completely destroy your parent strand. Okay, because then how are you rebuilding it? If you don't have the genetic information anymore, you've completely destroyed it, you can't duplicate it outwards. Okay, so that process doesn't work. So now we look at these two possible systems. What we have on the right, okay, is ultimately a completely new environment. Okay, the new genetic information is all entirely brand new and synthesized. So if we go through replication in that process, we maintain the parent DNA, and we end up with a completely new set of DNA as a duplicate. Or do we have semi-new? Okay, and what they're saying with semi-new is that we end up with a mix. Okay, one parent strand acts as the code, and the sister strand then binds into it. So you end up seeing kind of a combination of both of those. So the question becomes, well, which method is the one that is correct? Right, the only way we can really do this is to set up very careful experiments, and that was using density gradient ultracentrifugation. So if you remember, we briefly talked about that S symbol. We looked at ribosomes. There's the 50S, the 30S, the 80S. That S all had to do with density and centrifugation. Okay? So what they ended up doing is a very similar process with DNA, and they went through and took the parent strand, and they grew that under very particular conditions. Okay? We could grow that under radio conditions or radio labels. So all the phosphoruses, phosphori, the phosphorus atoms, okay, are all heavy atoms. Okay, meaning we can now detect a difference in density because a heavy phosphorus in the same volume is going to have a larger mass, so its density is going to be a little bit heavier. So what they went through and did is then let it go through replication with normal DNA. What happens? Or with normal nucleotides. Okay, what ends up happening? Well, it's going to synthesize, and we're going to end up with two new systems out of it. Okay? What would we expect to see? Well, in the, what did we call this? The semi-new system, what happens? You end up with two sets of DNA with equal densities. So if we went to centrifuge that, we'd really only get one isolation or one spot if we looked at it as kind of a TLC plate. Okay? If we look at the completely new, what happens? We'd end up with two. One where it's all heavy and one where it's all not. Okay? We could then continue this process because maybe it's just a fluke. So you could run this cycle multiple times over, and you can then end up starting to predict what happens afterwards if you're following a semi-new versus a completely new process. As you might guess by this figure that your textbook has put together, which process wins? The semi-new. Okay? So what ends up happening is we cleave or we open up the DNA, and then we bring in new nucleotides to match. We generate two strands of DNA. Okay? And that was all done through your density ultracentrifugation. You may hear reference to a name associated with that experiment, and I don't remember what the name is. I think it's Henson. I think there's a double S in it.
Mendel and Stahl or something?
Okay, maybe not a double S. That sounds pretty right. I think. Yeah. So, DNA replication. So now we understand a basic idea of what's happening on those differences. What do we need to go through to replicate our DNA? So what has to happen? Let's come up with some ideas.
The parent strand has to split.
Okay. We need to split the parent strand. Okay. So how am I going to split this double helix? Push a little bit further than that. So let's take a look at the model we've got in back. Okay, this is where it's going to be a little. We're also looking at an unwinding process. Okay. Which, if you remember back to that last conversation, it's a little bit worse than just unwinding. Because if we look at our DNA, we do have that double helix. But then what happens? Okay, if we just had our helix, we now have this giant strand of spaghetti string, okay, that's 6 feet long. Our cells aren't 6 feet long. How do we get that DNA strand actually to fit within the cell? You supercoil it. So you take that strand and you wind it even tighter. Okay, so not only do we have just the unwinding of the double helix, we also have, good thing I left myself lots of space. Okay. We also have a supercoiling unwinding that we have to worry about. Okay. So we have to do this at the beginning. What are we going to do at the end? You're going to have to put it back. So not all are we unwinding to begin with, we also have to unwind at the end. Or sorry, wind, rewind. Rewind? That's, we'll include the dash. Rewind at the end of the process. Don't date yourself too much with that one. Okay.
So it's two separate processes you're saying, unwinding and then supercoiling?
Does it have to be,
thing?
so, at that stage we're looking at how far does it have to unwind. Okay, so it's not quite two separate things. It is going to be done by the same process, but what we're looking at is undoing our supercoiling. So, undo supercoiling. Let's just call it that. Okay. What else has to happen? So right now what we've got is what we have in back. We have the ladder. What do you have to do?
Get those matching DNA nucleotides to complement or something.
So if we look at one possible process, here's the matching nucleotide. That it has nowhere to match. What has to happen before we can bring in another nucleotide? We now actually have to separate this. Okay? So we're now going to separate our DNA. Let's just say separate DNA. What else has to happen? So now we've got this separated. We don't want to quite say transcribe, but we now have to somehow get this to match up on the other side. Okay. So what ends up happening is we're going to have another enzyme that's going to say, okay, let's go ahead and put some two nucleotides together.
Isn't that transcriptase? Right.
We'll talk about different enzyme names in a little bit. Okay. So we now have our first nucleotide in. What happens next? Okay, keep going. So we can keep opening this up. Where's the next nucleotide?
It's not there yet.
It's not there yet. So what has to happen?
We have to build it.
It has to somehow get there. Right? So let's reach out and let's grab a nucleotide.
What if it's not the right one?
What if it's not the right one?
It won't bind.
Right? It's not that it won't bind, and so I spaced this a little bit oddly. It's not that it necessarily won't bind, but that hydrogen bonding interaction is relatively weak. What happens if it's weak? It comes on and it comes off. So we try again. Oh, look at that. I'm going to grab one. We now have a nice hydrogen bonding interaction. But now what happens? Has it now formed your DNA?
No, it has to attach.
It's still free floating. It can completely come away from the structure. So what has to happen?
It has to attach to the strand.
It has to attach to the strand somehow. So this needs to somehow link to the next piece.
Yeah, that's true.
What's the problem?
It's like the sugar base.
It doesn't match. So what ends up happening? Falls off again. Okay, and then what happens?
Tries again.
Tries again. Come on. Okay. And now, well, that wasn't supposed to happen. Okay. We can now potentially link up. Okay. And this process then repeats again and again and again. Okay. Interesting aspect of this is we were all doing this while we were watching it. Okay. So we can see what's going on. We can see that I built those pieces so they work properly. Do our molecules have eyes?
We don't know that yet.
Good answer.
Fair response, but let's try that a little bit differently.
Not that we know of.
Technically, they do have eyes. What is their eyes?
Well, they're chemical makeup.
Kind of. It's getting towards it. I remember one of the things that had to happen is we had to have these link up with each other. Why did they link up? Keep pushing that. Are intermolecular forces. Okay. So what's driving this process is favorable intermolecular forces that last for long enough that allow other chemical reactions to occur that then can perpetuate and allow it to stick and continue that process along. Okay. So not only are we then going through and matching our new nucleotides, but at the same time we have, that says matching, kind of a, repair is not what I was looking for. Error analysis? Correction? What's wrong with that?
Paper clips representing.
We'll talk about that in a second here. Correction slash repair mechanisms. So one of those correction or repair mechanisms is looking at just favorable interactions. So there's enough time in between that if we don't have a favorable interaction, it falls apart. Okay. What happens, oh, stupid thing, reconnect. What happens if, for some reason, it happened to do that next step pretty quickly? So let's see if we can do this. Pop that out. I think I sized these so that they would not link with each other. So there we go. There's a horrible interaction, which is so horrible it doesn't even want to stay up. We look at the next one as being a favorable interaction. What happens now? We now have a mismatch in the DNA. Do we continue replication?
Yes.
Yeah, which becomes problematic because now we have potentially an error or a mutation, which could lead to cell death, that could also lead to cancer, all sorts of other fun things. So what also has to happen? Once it goes through and replicates and puts in the new nucleotides, something else
And then it comes by and
needs to come back through and verify that everything is correct. So we get lots of these repair and processing issues. So we get our nucleotide insertion, which has some kind of correction repair built in, just based on the timeline it takes for it to go through and insert that nucleotide. But after we've passed it, let's call it replication, we're also going to have some repair mechanism. Each of these things that we've highlighted are then have to be functioned by a separate enzyme. Something else has to come in and process this. So when we're looking at replication or transcription or translation, these are vastly complicated processes with lots of moving parts that move in between. Okay? So unwind the DNA, separate our strands, select the correct base pair, add new base pair. Okay? Fuse the backbone, check for errors, fix errors, rewind DNA. Okay? Which are all the important aspects that we just kind of addressed. Each of those is pretty much run by a different enzyme with similar processes. For instance, if we take a look at unwinding the DNA versus rewinding the DNA, would we expect that to be a different enzyme?
Yes.
Okay, why?
Because one's going to coil and one's going to uncoil.
What if you just turn the enzyme around? You're probably looking at the same type of enzyme, but a different unit.
Different tail and different
Why does it need to be a different unit? Well, number one, one's at the beginning, one's at the end. Okay, so we're going to have those on at different processes. But it is probably a related, if not similar, enzyme. Okay. So the intention of this model was to have you guys kind of go through blindfold and attempt someone to go through and replicate that. If someone is brave enough to give that a shot, You are more than welcome to
what?
play with the model that we have in back. Okay, so before we go looking at movies.